Okay. A quick recap then as to where we've been with the nervous system. We started with an overview of the different component sections, if you like, sensory receptors, gathering the information from the internal and external environments, converting energy from one form to another really is their job. And what we should be on this morning is the eye, but because I missed yesterday, we will catch up with that next Monday. Out of the nerves in the middle there, we will look at the autonomic nervous system later on this morning. So I haven't put a ring round that, but there we go. We'll have a quick look at that. And then we will also, hopefully this morning, get on to the brain. So I'm being quite ambitious this morning in terms of the amount that I want to get through. And we'll see, yeah, well, you know, we will have time to catch up with it all, so I'm not too concerned. The next thing that we did was to start looking at different sorts of neurons. So we've got the difference between sensory neurons, motor neurons, and the multipolar connector neurons that you find in the central nervous system. So important thing with these was the myelination on them particularly. And when we got on to looking at the nerve impulses, of course, that was very important. We had a go at measuring your actual impulse speeds using your preferred and non-preferred hands. And curiously, there wasn't a significant difference between those. And then we had a quick recap on facilitated diffusion and active transport, because those were then going to become very important when we start looking at the way that a nerve impulse works. That's the resting potential and the action potential. We've got section then that takes us through the most complicated bit, and this does come up in the exam, but it is more of it's like a space filling exercise. So I give a description, but I'll leave gaps. So I might, for example, or I might give you alternative answers to choose. So I might say the sodium potassium pump delivers, and then leave a blank, sodium ions out of the cell. So you've got to remember the number 3. Or I might give you the option of the sodium potassium pump delivers 2 or 3 sodium ions out of the cell. So you've got to choose the right one. With these, remember, the way to remember it is that for the sodium ions going out, the number 3, so I put ... matches up with the 3 characters in a sodium ion, Na+, so 3 characters, and the 3 letters in OUT. So 3 sodium ions out, and then magically it also works the other way. So you've got the 2, the number 2, matching up with K+, 2 characters, and IN, 2 letters. So that exchange process is active transport. We also had to take account of the gated ion channels, which are for facilitated diffusion, and the fact that they weren't always open at the same time. We start off with the sodium channels closed, so the sodium ions couldn't diffuse in, but some of the potassium channels were open, so some of that could leak out. Now, the reason I'm recapping this mainly is because when we get on to how synapses work, we've got to look at how you go from this occurring in one cell to making it occur in the next cell, because it's not like having electrons flowing through an electrical cable. This is about a depolarization of a membrane. How do you pass that from one cell to another? So this is, you know, one reason for my recap. So this business about sodium channels and potassium channels being closed at this point or open is very important. We used a numerical description to show how the movement of these different ions could result in a difference that we describe as minus 70 millivolts, so that the inside has a charge which is negative compared with the outside. And then we went on to the action potential, and this is the thing that's going to need to be passed from one cell to the next at a synapse. So our next job is to look at synapses. And with the action potential, it's about the sodium channels starting to open. The sodium ions rush in, so you get more channels opening, more sodium rushing in, so that's positive feedback, one of the very, very rare examples of it. And it results in the membrane being depolarized, and you get this action potential of plus 40 millivolts, which means the inside is now positive. Now that is what you've got to pass on. When you've got two nerve cells, you've got depolarization occurring in one, you've got to make depolarization occur in the next one at this junction, and that's our next step. So it's where we're going to be going straight from this. The membrane has to be repolarized again before it can send another impulse along, and so we've got quite a lengthy description there on that. You have in your handout packs a sheet which I suggested, if you find them quite difficult to use, then find strategies like this where you look at a diagram that's on there and then you use a color coding sort of effect with a highlighter pen to show which parts of the diagram relate to which parts or which box, and the boxes are usually descriptive of what's going on in that particular part of a diagram. So that's we went through it using that. And that appears to be it. So that was as far as we got. Now, the next step then is to get onto synapses. So if you can have a fish around in your handouts, you'll find you've got a diagram of a synapse, which is fully labelled up. I'm just trying to find the heap of biology handouts on the desk. I think I've buried them somewhere. Oh, no, here they are. Okay, bear with me. But it should come straight after. No idea at the moment. I'm nearly there. You've got the sheet that's got the action potential on. Oh, yeah, it's straight after that. So it looks like this: synapse structure and function on one side, and alteration of synaptic transmission on the other side. All right. Now, the way I'm going to go about this is to draw my own diagram of this and sort of talk you through a description of what's going on as I do that. So it will come out with the same bits as you've got on the handout there, but I might sort of condense or not include some of the steps along the way, but the key parts are there. So synapses then is good job. And this is a matter of transmitting a nerve impulse, a wave of action potential, from one cell to another. Now, if we were looking at electricity passing through electrical cables, we'd be looking at a flow of electrons. And that's actually fairly straightforward to pass from one conductor to another, isn't it? But you have to have contact, don't you? I mean, very often pieces of electrical equipment don't work because a piece of wire's come loose, or contact has been lost. And the whole point about contact, think about what you know about metallic bonding from what we've done in chemistry in that bit of assignment. Think of your piece of copper, for example, in a copper wire, and you've got all your copper nuclei with the electrons delocalized around the outside, haven't you? And if you push some electrons in at one end, then some will fall out of the other end. Effectively, they shuttle. That's electricity. Well, that's a biologist's description of electricity, you've got to bear in mind. But it's actual subatomic particles, but they are moving. They are entities, if you like, that have to go from one piece of solid copper to another piece of solid copper. So even the smallest gap between them, and you can't move them along. So they have to make contact, don't you? So, in an electrical circuit, contact is everything. Otherwise, it won't flow. However, in the nervous system, a nerve impulse, of course, is not the flow of electrons. A nerve impulse is a wave of membrane depolarization. So you can't, it's not something that's flowing, it's something that's happening to the cell membrane. So we don't have contact, all right? There is no contact between nerve cells in a nerve pathway. And that always feels really odd. So you think about all the nerves in your brain and they're not actually contacting each other. How are they even there then? They're just floating. Well, there are a lot of other cells. There are cells like the Schwann cells that wrap themselves around to produce the myelin, and they actually sort of support all the cells. And these gaps between the cells, they are fractions of micrometers. I'm not sure how big the gap is now. It's not there in my brain, but we could find that quite easily, I'm sure. But the gaps between the cells are very important in terms of the transmission of impulses. It happens in a very distinct sequence, which you've got on the diagram in the handout, and I will go through it in pretty much the same sort of way, but I'll sort of talk around bits as we go. So what we need is an incoming nerve cell, neuron. So we look at the terminal of that incoming one, and then an outgoing nerve cell. So, it might be a connection, let's say, between a sensory neuron that's picked up some touch in your fingers, and then a connection with a neuron in your spinal cord, something like that. Okay, we need to have a way of transmitting the impulse between those two cells. So let's I'll just roll that off and make the whole board space, and then I'm going to sort of write round, sort of draw and write, put the writing around the outside of it to try and explain what's happening. So here is my incoming neuron. So this is my neuron terminal, and I'm going to draw things in it that are not necessarily obvious to start with. And let's make this a myelinated cell, so there's some myelin up there. And this incoming neuron, the correct term for something that's coming in to a structure is afferent. The one that goes out is then efferent. Okay? And it is important you get them the right way around, afferent and efferent, and it's a bit like affect and effect. They are two completely different words. Right? Affect is a verb, effect is a noun, usually. But afferent neurons come in, efferent neurons go out. So here is our, this is the terminal of the afferent neuron. And it's a very odd shape, the way I've drawn it. It doesn't matter what shape it is. It just needs to be a sort of slightly expanded part of the neuron. Right. Now, the two little dents in the bottom there, they are to do with exocytosis. So I'm going to be coming back to those. And that's how this cell is going to release certain neurotransmitter chemicals that we're going to come up against. Right, I now need to draw the efferent neuron, the one that's going out. And this is quite tricky because the membrane of that cell, it has sodium channels in it, these gated sodium channels, because that's what we're trying to pass on, is opening the sodium channels, isn't it? Because that's an action potential. And those sodium channels are attached to little receptors which, when they're stimulated, they will open the sodium channels. And the little receptors pick up the chemicals that are going to come from this afferent neuron. So they are difficult to draw in the space that we sort of have in a diagram, but just making them a bit bigger than seems reasonable. I'm going to make that that's a channel. Oh, yeah, I need to reset the board. And associated with it, I'm just going to put like a little cup shape. Oh, that's not as good as I wanted it to be. So a channel with a little cup shape, and then a channel with a little cup shape. And that is our efferent neuron. You can refer to these as presynaptic membranes and postsynaptic membranes. That's quite useful. But for the time being, I'm just going to put this as the efferent, oh, it's the whole thing, not just the surface. So this is our efferent neurone. I'll put membrane. Right, yeah, those little things are not quite what I was intending them to look like. They would have been better if I could have gone membrane and then perhaps channel. Or something like that works a little bit better. I've got them sticking out too far, but you get hopefully you get the general idea that we've got a channel that is attached to a little receptor. I would try and rub out a bit of them, but we know what's likely to happen. I don't want to redraw them all at this point. Okay, so those are the two cells. Oh, I've some other bits I need to put in here. One of the most important processes, of course, for setting up the resting potential is... Sorry? Yes, we need the mitochondria because they are going to produce the energy for the active transport. That's the process setting up the resting potential, the sodium-potassium pump, so we'll have a load of those. I hardly need labelling, but I suppose we should, shouldn't we? So we've got mitochondria in there. And then the other things that you can see on the diagram in the handouts is you have a lot of little vesicles. Now, vesicles are bits of membrane, and that's what these two things are here. All right, they're bits of membrane that enclose chemicals that have been made in the cell, and these are going to be released. I'm going to use another colour just to make it obvious, so I'm going to just put some red dots in there, which is quite tricky when your board's not lined up. They end up looking a bit like chocolate chip cookies. And these are what's going to come out here. And these, of course, are going to end up binding to these structures. So it's those little red dots that are going to cause the sodium channels to open. And when the sodium channels open, sodium ions will rush into that efferent neuron, the postsynaptic cell, and they'll depolarize that membrane and the action potential is passed on. But we need a little bit more before we do that. I do feel I should write postsynaptic and presynaptic cell on there, but I haven't really got room. Let me put it in here. Presynaptic cell or neuron. I'll put postsynaptic there. Pre always means before, doesn't it? And post means after. So they're quite useful words. Right, now we're going to look at the steps that occur in this process. First thing is, of course, you've got to have an action potential arrive here. So that's a wave of depolarization that reaches this terminal. And it will arrive at this uncovered, because we've got the myelin up here. You're not going to have the action potential there, but you are going to have it on, this part of, I didn't really want to make that thicker, just wanted to go over it. But that part of the membrane and this part, remember it's a circular cross-section. You've got a three-dimensional structure there. So you're going to get that particular part of the cell being depolarized. The effect that that has when it arrives at this terminal is to open calcium channels. Now, I haven't drawn calcium channels in here. If you have a look at the handout, I suppose I should do this sort of alongside, shouldn't I? So number one, the box at the sort of top right of the labels there, increase in local calcium ion concentration. And what that means then is that depolarization of the membrane at the synaptic button, they describe it as this little bit on the end. We sometimes used to call it synaptic knob, but for some reason they've stopped saying that. They object to teachers saying knob in a class, and I always thought it was a good opportunity, really. But, it's not a word you get to use very often. But this, I just call it a terminal. So we've got the calcium being able to flow quite quickly into this terminal from the surrounding tissue fluid. It's what we've got around there. So the first thing then is, and I'll do it in a similar way to that, but without quite so many steps, I think, and I'm going to go the other way around because I haven't got any space on the right-hand side. So my first step would be that and I'm just going to put AP for action potential, arrives at the terminal opening Ca2 plus channels. So ions flood in. The word flood is just a representation of the fact that this is really rapid and large scale. So a lot of calcium rushes in to that terminal. And it's one of the important uses of calcium in the body. We tend to think, if we say, oh, why do you need calcium? Most people would say, yeah, teeth and bones, because it actually forms a physical part of the chemicals, the calcium phosphate in teeth and bones. That's the thing that first springs to mind. But we also need calcium for nerves to work. And there's a sort of, there's a homeostatic control of calcium levels in the blood. And it's done via hormones. There are two hormones involved. One's called calcitonin. The other one is the parathyroid hormone, and I can never remember which one does what, but one of them raises your blood calcium by releasing it from your bones. The other one lowers your blood calcium by putting it back into your bones. So, if you end up with too much coming out of your bones, of course, that's what can cause osteoporosis. Brittle bone disease, there is a misunderstanding that those are the same thing. Brittle bone disease is a genetic condition that people have from birth. Osteoporosis is a loss just of the hard chemical. Tends to come with age, particularly in women. But I'm sidetracking now, aren't I? But it's all about getting the calcium levels up in the blood. Now, not only do we need it for nerves, we also need calcium for blood clotting. So low levels of calcium will actually affect the speed of blood clotting. So it's really a multi-purpose element in the body. We're not concerned with all that other stuff, though. That's just extra. You won't need that for the exam, but you might need this. So the first thing then is our action potential. Then it's that wave of depolarization of the membrane gets to this point on the neuron. That will open calcium channels so that, I could put little arrows, so you've got, calcium is going in here. All right, so that's happening there. Now, the effect of that is that it makes our vesicles in here, which contain a substance that's just generally called neurotransmitter, it makes them move to this presynaptic membrane. Okay? So that, unless that calcium floods in, they don't do that. So vesicles of neurotransmitter, and I'll come back to the neurotransmitter in a bit, but vesicles of neurotransmitter move to the presynaptic membrane. Now, the neurotransmitter has to be released by the process of, yes, I heard that, exocytosis. So it's where the vesicles fuse with the membrane and just release their contents. So it's one of the few examples that we actually meet in this course, but we are working our way through most of those transport processes that you did in the first unit. They probably are the most important bit of that in terms of moving on. So we've had facilitated diffusion and active transport. Now we've got exocytosis. Ultimately, when this has been used, it has to be taken back up again. So could be endocytosis. Yeah, not necessarily. We can have special transporters for it, but yeah. So the neurotransmitter is released, and it's released into this gap, which is known as the synaptic cleft. All right, a cleft is like a channel, isn't it? And if I'm keeping track of sort of what's happening here, these I'm going to talk about the substances that actually are examples of neurotransmitters in a bit. Let me just move this up so we can all see what I'm doing. So our next bit, the third bit, is that the neurotransmitter is released into synaptic cleft by exocytosis. All right, so that's what's happening up here. If you run out of space, just put numbers and then write the labels separately and refer to the handout one as well. They diffuse, and that would be simple diffusion really, I suppose, very, very rapidly across that cleft because it's only tiny. You know, we are talking at the micrometer scale here. And so once it's diffused across, it binds itself to one of these little receptors that's attached to a sodium channel. Now that's not very obvious on the diagram that you've got, but it is quite difficult to draw, I suppose. But you know, they seem to have separate little square receptors for their neurotransmitters, and then the sodium channels are separate, but they would be linked in some way. So what's happening here then, my fourth step, and this is the important bit, isn't it, if you think about it, because this is where we're actually trying to pass the action potential on. So the neurotransmitter binds to, and it will be a receptor that's got a shape to fit that neurotransmitter, so we describe it as stereospecific. Yeah. Some neurons actually have different types of receptor on them, and they have several incoming neurons. And you can have some neurotransmitters which will cause an impulse to be passed on, and some that will inhibit an impulse being passed on. So you can have inhibitory or excitatory synapses, effectively. So it's a point at which you can control what's going on in the nervous system. So we've got here, we'll just leave it at 1 because otherwise it just gets too confusing. So the neurotransmitter binds to stereospecific receptor. Sure, I'm going to have room for everything. Let's see how we go. Associated with, that's a long word. Can I have something smaller? I'd love to do. Associated with sodium channels. In which case I'll have to have an S there. Oh, why didn't I just put Na+? Make it harder for myself, don't I? Associated with sodium channels in postsynaptic membrane. And then I've got to continue on this same one, really, because, well, let's link that to that. Whoops. I suppose I could put a fifth one. The sodium channels will then open, sodium ions rush in, and the postsynaptic membrane's depolarized. So I'll put that as a separate point, but because it doesn't happen at the same time here, although it will be very closely related, won't it? So number 5, which I can't really label up there with a line. So Na plus channels open. So ions rush in and depolarize postsynaptic membrane. So there we go. We've managed to pass on our action potential. However, we've got a bit of a problem here. Do you want that neurotransmitter, morning, to stay in the synaptic cleft? You don't. Sorry? No, it just needs to be removed. We need to keep taking it away, because otherwise it'll just keep firing off impulses, and we don't want nerves to work like that, do we? So how the heck are we going to get rid of it, and what are we going to do with it? Well, actually, we need to reuse it. We can't afford the resources in cells to just keep making stuff and then getting rid of it. You know, we've got to be fairly conservative about resources. So the final thing that happens here would be that the neurotransmitter is actually broken down possibly by an enzyme. There's one very common one I'll give you here. May not be broken down, but it will actually be reabsorbed into the presynaptic cell. So the neurotransmitter, and I'll just put maybe broken down by enzyme, and the name I'm going to give you is acetylcholine esterase, if I can fit it all on. Yes, I can. That breaks down a neurotransmitter called acetylcholine. Acetylcholine esterase. Once it's been broken down, it's then in parts that can be reconstructed by the cell, and some neurotransmitters don't even get broken down, but they are taken back into the presynaptic cell and reused. So it may be broken down by the enzymes such as that, and are reabsorbed for reuse in the presynaptic cell. Right. That then describes the process. Now, there are probably about 50 different neurotransmitters-ish. A lot of them are very closely related to each other. Some of them you may have heard of. Most of them I haven't heard of. But, you know, we don't on a daily basis. I've given you the name of one there, acetylcholine. So that's the most common one, and that's the one that just tends to work in our sort of somatic nervous system, controlling our conscious sort of responses. Now, there is a different neurotransmitter that works in our autonomic nervous system, which is the one that works sort of below our level of consciousness. It's a homeostatic sort of system, controlling things like heart rate and stuff like that. And there is a neurotransmitter that has a very similar name to a hormone. And it's a hormone that could, for example, speed up your heart rate. What hormone would do that? Adrenaline, yeah. And the neurotransmitter works in exactly the same way, and it's called noradrenaline, and it just has a slightly different structure. So adrenaline and noradrenaline are pretty much the same thing. But adrenaline is a hormone, noradrenaline is a neurotransmitter. It's just that it's a neurotransmitter that works in the part of the autonomic nervous system that has the same effect as adrenaline. So we've got acetylcholine, we've got noradrenaline, or if you were reading American sources, now anybody listen to the podcasts yet? Yeah? So what would it be called in America? I did say on there, yeah. yeah, from American sources, and I don't want to see the American word in assignments. If you choose to do this one, it's epinephrine. Yeah. Now, the weird thing is, you see, the two words mean the same thing. Adrenaline means ad means next to, and renal means kidneys. Yeah. So the adrenal glands sit on top of the kidneys, don't they? If you think back to what we did on hormones, and adrenaline comes from the adrenal glands. Now, the American word epinephrine, well, epi means on the outside, and nephro is to do with kidneys. So epinephrine and norepinephrine, same thing. But epinephrine means exactly the same thing. However, I think they still talk about the adrenal glands. So there's a bit of a mismatch there. If you say the adrenal glands produce epinephrine, then you're mixing two bits of terminology, aren't you? I suspect the words have got different roots, whether they're sort of Greek or Latin or whatever. But yeah, so hence, if somebody is likely to suffer a severe allergic reaction and anaphylactic shock, they carry a EpiPen. And the epi is for epinephrine. And so they carry an EpiPen which delivers adrenaline, or in America, epinephrine. It's exactly the same thing. All right, but I don't know what we'd call it if we invented it in this country. An AddyPen, it doesn't work, does it? But an EpiPen is so called because the epinephrine origin. Anyway, sidetracking a bit there. That's two neurotransmitters. So you have acetylcholine, noradrenaline. We also have some others that you might have heard of. Let's think a few examples. I'm trying to think ones that might have come up. They work in different ways, in different parts of the nervous system to give us control, really. So we've got acetylcholine. We've got noradrenaline. Anybody know any others? You'll have heard of some of them, I'm sure. Heard of dopamine? Yep, the dopamine is a neurotransmitter. Now that has two jobs in the nervous system. There is what they call a dopamine reward pathway in the brain that uses that particular neurotransmitter, which is associated with addiction, actually. It's the pathway is designed to reward us for doing things that keep us alive, like eating and drinking. So they make you feel good, so you do it again. All right. Otherwise, why would you do it? You don't have this drive because it keeps you alive. So the dopamine reward pathway rewards certain types of behavior, and the pathway can be triggered by certain drugs and things like that. So it's one of the pathways and one of the mechanisms that's involved in drug addiction, particularly. The other thing that dopamine does is it's produced in a completely separate part of the brain as well, and it helps to coordinate voluntary movement. And there is one particular neurological condition where the cells that produce the dopamine that controls movement die, causing problems with movement, which is Parkinson's. Yeah? I could see that wasn't on the tip of anybody's tongue there, but Parkinson's disease is about initiating voluntary movement. Somebody with Parkinson's, you usually associate it with tremor, don't you? It's not so much, that's not really the issue. It is about actually starting the movement, and without sufficient dopamine, people with Parkinson's get frozen in position, can't stand up, and that sort of thing. And so consequently, one of the main treatments, one of the main pharmacological therapies for Parkinson's is to provide dopamine. It's a very sort of inaccurate, unpredictable sort of treatment, but you just give dopamine fairly regularly, you can get some control back over movement. Another one that I bet you've heard of is serotonin. Yeah, serotonin is it's a brain chemical really that helps to maintain um, it's just a sort of modulating sort of chemical that maintains control and mood, if you like. They often call it a mood stabilizer, and people sometimes suffer from a lack of serotonin. Reason being that the brain cells are a bit too ready to take it back up sometimes. They don't leave enough serotonin in the brain to keep mood stable. And so consequently, there's a class of what are known as antidepressants, but shouldn't really be. They are more accurately mood stabilizers, and that's the SSRIs, the serotonin selective serotonin reuptake inhibitors. So now you think about that. I can't remember which way around the S's go, but serotonin selective means that it's just about them. Reuptake inhibitors, so it stops the brain cells taking the serotonin back up, allows it to be left in the synapses, and allows a more stable mood. And very often it's just about people being able to regain control over things that have, been not so. There is another one that you could come across. Depends. Sorry? That's not oxy. I can't think of any that start with oxy now off the top of my head. The one I thought people might have heard of is known as GABA, which is gamma aminobutyric acid. If you can find an oxy one, yeah, there may well be others that people have heard of, depending on if you've got friends, relations, or your own experience maybe of anything neurological, whether it's mental health issues or musculoskeletal problems. Sometimes certain drugs will target the way that these work, and that's the next thing we're going to look at. So that's about 10% of the neurotransmitters that we've got rattling about doing various different jobs. So they are, as I say, in different parts of the system, and they provide these different mechanisms for controlling response. Oh, endorphins. Yeah, sort of. And they have specific names, the endorphins. We're going to look at those in a minute anyway. I'm not sure that they're actual neurotransmitters, but they do modify the way that synapses work. So if you turn over the page, on the other side, you've got alteration of synaptic transmission. And on this side, you've got a whole load of different substances that can change the way that synapses work. Now, you don't have to know any of this, but it's interesting, and it gets you sort of a bit clearer on what synapses are actually doing. Right, back with it. You've got so many different aspects to the way a synapse works. You've got the production of the neurotransmitter, you've got the release of the neurotransmitter, you've got the receptors actually being available to bind with it, you've got the opening of the sodium channels, you've got the removal of the neurotransmitter once it's been used. And a lot of chemicals are actually designed, if they're drugs, they're designed. If they're things we've just happen to have come across in nature, they're not necessarily designed to do this, but they can interfere with the way that those processes work. So we're going to have a look at a few of these. I'm just going to skim through these because I don't want to spend ages on this, but you can obviously sort of read through it yourselves as well. If we start, I don't know which side to start. Let's start on the left-hand side and work sort of anti-clockwise. You've got a box on the left there that says limited removal of neurotransmitter. And what this is about is leaving the neurotransmitter in the synapse. So that's like what an SSRI drug would do. So that can affect the way that synapses work by making the serotonin more available. But they've got some examples in here. For example, cocaine. Cocaine acts as a euphoric drug by inhibiting the reuptake of noradrenaline. All right, so it will leave adrenaline in, effectively, or noradrenaline in synapses. And you think about the effect of adrenaline, it's an excitatory sort of substance. So cocaine is inhibiting the reuptake of noradrenaline. It's leaving noradrenaline, so it raises the level of sort of activity and awareness, alertness, and all that sort of thing. And then, if we just skip over the bit about weed killers, we've got amphetamines, inhibit the enzyme monoamine oxidase, which degrades adrenaline and dopamine. So those are stimulants as well. So, you know, there's examples where the neurotransmitter's not being removed at the end. The next one down that side, mimicry of neurotransmitter. Nicotine at low concentrations mimics acetylcholine, so that's a stimulant, isn't it? There's some others there, but then the bottom bit of this is quite interesting. Endorphins, such as beta-endorphin and enkephalin, are endogenously produced peptides. That means they're like proteins produced in cells, which mimic the opiate drugs, such as heroin and morphine, and act as natural painkillers and euphorics. Now, to me, that's actually the wrong way round. That's actually been written the wrong way round. I would say that the drugs mimic the endorphins, yeah? But those opiates have the same sort of chemical effect as our natural endorphins, and endorphins they're the chemicals that are produced in response to stress, very often to keep the body going. The classic example is people who do a lot of really high intensity sport and gym, people who go to the gym every day, that sort of thing, they have to do it. They actually get addicted to their own endorphins and get that sort of high off it. And a lot of people who perhaps go running and that sort of thing will get to the point where, yes, the body's trying to protect itself actually, but they'll do it because they get a feeling of pleasure from the endorphins. And you've got, there's various ways of getting this good feeling. So you've got intense exercise. I mean, they're there for that, so that if, for example, you go back into our history, if you were running away from a wild animal, you fell over and twisted your ankle, you can't just roll on the floor like a footballer, waiting for the wet sponge to come out and make you better, can you? You've got to get up and keep running. So, yes, there's an interesting side to evolution there, isn't there? So the endorphins are that. But you will hear of sportsmen sometimes carrying on, even with broken bones sometimes, don't you? Broken bone in the foot or something. But they carried on till the end of the game. Then come the end of the game, of course, then they're limping off. And but it's the endorphins that keep you going. So you can do the intense exercise and get your endorphins. You could take heroin. The other option is chocolate. Yeah. Now chocolate, it's on a much, much lower scale, but there are chemicals in chocolate that do a similar thing. All right. So, between us, all right, you got a choice. You got intense exercise, heroin, or chocolate. Well, you can tell I'm the one that does the intense exercise. I don't know about the rest of you, but yeah, in terms of the chemistry of the brain, that's what's going on. They're working on the same sort of system. So it's a mimicry thing. We have the endorphins, but and this is why chocolate makes a lot of us feel good. Some people don't like it. I just don't get that. But yeah, it's a neurotransmitter thing. Just coming round the bottom there, sticking with the label boxes, inhibition of adenyl cyclase. Now, don't know if you remember, I mentioned that was in the second messenger system in the peptide hormones. So adenyl cyclase is about producing a second messenger. So if you inhibit that, you can prevent things happening. So, or it actually leaves the second messenger running, and caffeine inhibits that. So hence that's a stimulant. Coming around from the bottom there, altered availability of postsynaptic receptors. So those little receptors attached to the sodium channels can vary in their availability. So things like curare, a poison used by South American Indians to tip the heads of arrows and darts, competes for acetylcholine receptors. Yeah, a big problem we have around here, isn't it? Also some hypnotic drugs and very high doses of nicotine can do that as well, and so that's actually it's lethal. Coming round and up that side, decreased sensitivity of the postsynaptic membrane. Certain depressants, barbiturates for example, reduce the ion permeability there of the postsynaptic membrane, binding to the sodium or potassium ions. Actually binding to the ions, that's interesting. And then the box above that, reduction in release of neurotransmitter, so just reducing the amount of neurotransmitter that comes out. And one example of something that does that is botulinus toxin. What's that used for? Take the first, sorry? Botox. Yeah, Botox, botulinus toxin. Yeah, comes from a bacterium, botulinus. I'll try to think what its other name is. Can't off the top of my head. But it causes a fatal form of food poisoning, botulism. And the reason it can do that is because it is toxic, potent in very small amounts, and yeah, so it does make you wonder why people submit themselves to having it injected into their faces and things, doesn't it? But it does. If it's localized, it's not an issue, but potentially quite dangerous. It is also used for other things. And it's associated with nerve impulses. One example is in people who suffer from the real extreme sweating. People whose palms practically, you may not, but people can have such sweating problems their palms practically drip all the time, and armpits, just constantly wet. And if you inject those nerves with Botox, it stops the problem. It has to be done every few months, but it's an absolute sort of social lifesaver, isn't it? That sort of thing. The only other option is actually to cut those nerves and to actually find them. Sorry? Oh right, what, having them cut or yeah? Yeah. No, I think they used to cut them, and I think now they find all right, cutting them is permanent, but Botox is a lot more. It's less sort of terminal, isn't it? You can't bring them back. The other use, and I've not heard of this a lot, but it came up a few years ago, so I always mention it, is that it has potential use in young children with cerebral palsy. Because one of the problems that they have, you might be aware, is that they're very often up on their toes, and it can make learning to walk really difficult. And one of the reasons for being up on their toes is that the muscles and nerves up the back of the leg, you've got to pretend my arm is a leg, are very tight. And so if you inject into appropriate nerves there, you can actually get the heel to drop. And so, the thing is with this, I think it can only be used a limited number of times, or it doesn't work in everybody as well. That it depends on the reasons for the disability. But if you can, in the first few years of life, get the heels to drop, they will learn a better walk early on, and yes, eventually the tightening may return, but they will have better muscle development and skeletal development. So it's a worthwhile therapy to try and improve walking. So you've got a heck of a lot of potential targets in synapses for different chemical compounds to actually interfere. And so, you've got this, amount of neurotransmitter mimicking the neurotransmitter, availability of the receptors, clearance of the neurotransmitter. So, it is fascinating. I think the nervous system and applications of the nervous system are, potentially very interesting in terms of, how you can apply it. And so there we go. That's synapses. Right. Now, the next job is to move on to the central nervous system, but I don't think we're going to get very far with that. So we'll start with just a little overview to get ourselves started, and then we'll probably take a break and come back to it. I don't know. We don't need to do very much on it. As far as the central nervous system is concerned, you do get a question in the exam on this, and it really is only about parts of the brain and what they do. But we need to sort of work ourselves up to the brain. Well, that's what I have done in the past anyway. That's not to say it will be exactly the same. So the central nervous system is now one of our actual component parts. What we've looked at up till this point are the building materials, really. And I think all we'll do for now, because I know I'm going to have to pop down to reception very shortly, is really just to sort of set the scene with this, and then we'll take a break and I'll come back to this properly, so we'll have a slightly early break. But the central nervous system then is the brain and the spinal cord. And we'll just have a look briefly at the development of the brain, and then we'll leave the parts until after we've had a break. If you have a look at the next handout along, you've got two sides there. You've got one side that takes you through the development of the brain, so I'm just going to have a quick look at that. The other side is about the parts and what they do. There's a piece that I've got further on in there about body temperature. I may not get onto that. It doesn't matter particularly. But just having a look at this bit about the development of the brain. We tend to think of ourselves as having very highly developed brains, don't we? But it really is only an expanded front end of a spinal cord, or a neural cord, really. There are lots and lots of very simple animals that have a nerve cord running the length of their bodies, and perhaps a few cell bodies at one end that help to detect light and stuff like that. But nothing that you'd call a brain, and it is quite difficult to define, you know, where does it become what we'd call a brain. And so, you know, if you've got an animal with a nerve cord, then evolutionarily, you know, you might then start to get perhaps a few extra cells developing at the front end, and then, you know, this gets bigger and bigger until you end up with, you know, what we would call a brain. And then of course put it the other way up, and, you know, you've got a vertical animal like ourselves. And if you look at the diagram, well, you've got two diagrams that I want to refer to here. You've got the one at the top, which shows how this develops from, if you start off with your little ball of cells that develops from a fertilized egg cell. All right. We would start to see this. It tells us we're about 16th day of development in humans. Obviously, other animals different. You start to get an area on the top of this ball of cells, and the ball of cells knows where, or the cells at the top know that they are there, because you start to get chemical gradients developing just by virtue of position, really. And this little area, it's called the neural plate. And you've got to think of this three-dimensionally as a little ball. You then start to get a little dent sort of occurring in there. Oh dear, that's supposed to be a bit rounded. And as this ball of cells develops into sort of more of a sausagey shape, you know, elongates, because, you know, that's what we end up as, then you start to get this dent being much more defined. So that's what the diagrams are on there, until running through our elongated sausagey shape as a cross section, you actually have a tube. All right, that's a neural tube. And early on, a lot of animals have these. Now, sometimes that doesn't form properly, and the top doesn't close over properly, and it causes spina bifida, yeah, otherwise known as a neural tube defect. And in some people, and it can actually be lethal. If it doesn't close up at all, then it really is lethal. But people with spina bifida, and it's actually really rare these days, have, when they're born, the spine isn't properly closed in, and you can see exposed spinal cord. Surgery can actually close it over, but the nerves will be damaged, and they usually have some degree of disability below that point. And it's the reason why pregnant women take folic acid. That's the one. Yeah. Yeah, well done. Well done. But yeah, folic acid is known to encourage the proper development of the neural tube. So it's supposed to be taken sort of, in preparation for pregnancy and during pregnancy to help that close up properly. Hence, I don't know if it's added to anything at the moment. They've been talking. Yeah? Oh, I don't know. Yeah? I don't know. Oh, right. Yeah. Well, I know that they keep talking about adding it to bread, but there is this problem with adding anything to food that not everybody needs, because people will object to it. Plus, of course, it's going to add to expense. So but yeah, folic acid is involved in this development of the neural tube anyway. And then what happens in humans, mammals generally, if you look at that little series of 9 diagrams at the left there, going from 3 weeks through 4, 5, 7, 11, and then up to 4 months, 6 months, 8 months and newborn, you can see the development of a brain. And it gradually gets bigger and bigger, and it folds over, doesn't it? So, you start with something, if I take my little diagrams from over there, you start with something sort of like that, but then it sort of folds over a bit like that, then it folds over a bit more, and, you end up with this thing that turns into a brain. And the further you look at the development, the more texture you can see to the surface of it as well. And those crinkles that you get on the surface of the brain, and I'll get the model out after break. In premature babies, their brains don't have those. Those are part of the development, and it's a sort of increasing surface area thing that, the further the development goes, the better those, I can't remember what they're called now, but the crinkles on the surface of the brain, the better developed those are. Okay, well, different bits of the brain have got different jobs, and I'm not going to start that now, so we'll take a break at this point, and then we'll come back to our brain bits after we've had some caffeine, maybe. So go and interfere with your synapses. Okay. Now, getting back to the brain, then you've got this sheet with the developmental diagrams and things on it, and then at the bottom of that page you've got a diagram that is not ideal. I just stuck it in to fill the space, I think, really, showing that there are these different parts to the brain. Now, what I need to do is go and fish a brain model out of the cupboard, so bear with me. I realise this will sound very strange on a recording. Okay, the brain model. I used to have this out on open evenings as a bit of a challenge to people. You can take it apart very easily, but getting it back together again is a nightmare. The brain is a very three-dimensional thing. Like all of the structures that we've been looking at, you've got to think in 3 dimensions, and you only get diagrams that are 2-dimensional. So it's really difficult sometimes, because you get the impression that everything is just laid out side by side, and that the shape that they have in 3 dimensions, it's just not expressed in diagrams very easily. So I will take this model apart and wave it about while I'm talking about the brain. But the first thing I think I need to do is to draw you a diagram, a simple diagram, to just locate the main parts. What we have, put it the same way around as that one. Okay, a diagram of a head. I need it to be slightly better than the one. Oh dear, that's not very good at all. It's amazingly difficult on this board to get the shape right. Oh, a detached nose. Never mind. There we go. Yeah, not one of my better head shapes, that one, I must say. I'm sorry, I've got to redo the nose. The nose is completely irrelevant to the brains. Yeah, okay. Not quite the shape I was looking for. It'll do. Obviously, inside the head, we've got the skull. The skull has got 2 parts to it. Right? We have the brain box, right, or cranium, and we have the face stuck on the front of that. So the facial bones are on the front of the cranium. We're not concerned about the facial bones here, the ones that form the eye sockets and the nose and the jaw and all that. But we have the skull coming round more or less like that, and then a connection with the spine. And I've got the spine starting a bit high up there, so I'm going to bring it down a little bit. But the spine consists of bones that have holes running through them. So, you know, I'm leaving a channel running through the bones here, and these are just cross sections through vertebrae. Then inside the skull, we've got the brain, and the bit that we tend to think of as like the brain in a bottle in a horror film or something like that is this crinkly bit, and it's known as the cerebrum, or the cerebral hemispheres. And that sort of fills the main, shape-wise, this is appalling, sorry about this, that's going to fill the main sort of volume of the skull, like that. Yeah, hence it's quite useful for you to have a ready done diagram of this. We then have a lot of other sort of internal bits that we need to be able to identify. Now, as well as the cerebrum, there is another bit at the back which is known as the cerebellum. Now, that, on the diagram that you've got over the page, it looks a bit like a peach, but I'll show you the sort of shape of it in a minute. And actually in cross section, it's more likely drawn sort of more like a leaf, like that, with a line going through it. And cerebellum just means small brain. All right, so cerebrum is the main part, and then cerebellum is the small bit that's attached. We have in the central area, and I'm not being very accurate with this at all, there's a bundle of fibres that connects the two hemispheres called the corpus callosum. Talk about that in a minute. And then we've got, how best to sort of put these bits on. We've got a bit that we've actually met, the hypothalamus, pituitary gland, and some other bits, let's just say. All right, that's keeping it very vague at the back there. Oh no, sorry, I've not joined it up properly. I had a feeling I'd got a bit missing there. Hang on, need to join it up. Otherwise I haven't got a connection to the spinal cord. So I'm standing too close to it, I think that's a problem. Right, spinal cord should come down there. And so, that's, yeah, all right, that makes it look like the cerebellum's just dangling. Sorry, that's one of the worst brains I've ever drawn. I should have just got one off the internet, shouldn't I? Right, this is all connected up, so I'm not going to try and change the diagram now, but the parts, yeah, I can't believe how bad that is. The parts, in terms of their location, are pretty much okay. So we have, up here, we have the cerebrum, or cerebral hemispheres. And we have a left and right cerebral hemisphere. And the two sides, you might be aware, control the opposite sides of the body. Yeah, all familiar with that idea. So left controls right, right controls left. There are some bits where you get crossovers. So for example, eyes, you get information from each eye going to each side. There is the few fibres cross so that you actually get both eyes getting information, both eyes sending information to both sides of the brain. But generally speaking, there are different different sorts of emphasis on the two sides in terms of what they control. So you talk about people sort of being left-brained or right-brained sometimes. I can never remember exactly which bits go where, but, you know, some people perhaps are more emotional, some people are more logical, some people, you know, those sort of things are controlled by the two different sides. And for some reason, that's something that just doesn't stick with me as to remembering what the difference is. We get the same areas located on both sides in terms of things like sensation and motor control. But other aspects can differ somewhat between the two sides, and things like, I'll give you an example, communication. It's possible to map out what happens where in the brain when we communicate. And communication is about listening and responding and that sort of thing. And it's possible to work out which side of the brain is doing what, when we're being spoken to and we're having to answer questions and that sort of thing. And the way that this is mapped out is by using medical scanners, where you can use functional MRI, magnetic resonance imaging, which can actually pick up different sort of things going on in the brain, or you can actually use radioactive isotopes incorporated into glucose. Glucose with a radioactive isotope of oxygen can be used because the brain will only use glucose, and it's got a very, very short half-life, so it doesn't cause any damage. It's, you know, almost as soon as you've got it, it's gone. And if you give somebody this radioactive glucose and then ask them to do things and then image their brain, of course the bits of brain that are using most glucose will light up with these little areas of radioactivity. And so you can find out which areas are being involved in communication. And one of the things that keeps being flagged up, sort of, you know, and there'll be studies on this all over the place, is that there's a difference between males and females in communication in the brain. That with males it tends to light up on one side only, whereas with women we've got bits lighting up all over the place. And it is that idea to some extent of the multitasking versus focus. Yeah, I'll have to sidetrack onto that in a minute as well. It's not a bad thing at all. You know, the fact that male and female brains do work slightly differently, but there is a whole continuum. It's not like, oh, male brain, female brain, that's it. It's like there's a whole scale, and, you know, we're somewhere on that scale, and, you know, you might have a brain that's more like the other sex. It doesn't mean anything specific. It's just, you know, that's the way the wiring works. But the cerebral hemispheres, these have what I would describe as three main jobs. You can map out areas of sensory input. You know, you can point at a bit and say, that is the area that deals with incoming sound, or that is the area that deals with the sense of touch from the tip of my left little finger, or my right big toe, or the middle of my back. And you can map it out. There are absolute regions where those are located in the cerebral hemispheres. Similarly, motor control has very, very definite regions, all right, that you can say that is the area that controls me bending my, well, whichever fingers, you know, happen to be. It's very difficult to do your little finger on its own, you know, but one particular area of the brain controls that, another area controls that, and it's the same in everybody. So sensory input, motor control, but then of course what we associate most with the cerebrum is all the higher mental functions, don't we? Because we have very developed cerebral hemispheres, particularly the frontal lobe, which is the bit, you know, that we you know, we locate at the sort of forehead area, and the higher mental functions are things like memory, emotions, behavior, or all those sort of things, you know, that we associate with being more advanced, if you like. So quite significant. You know, this is what we think of as our brain. So we've got sensory input, we've got motor output or motor control, and then these higher mental functions, this is where all the processing goes on. This is our ability to think, if you like, reasoning, that sort of thing. You know, the sort of things that sort of separate us from other animals, largely. You know, although obviously, you know, the closer related other animals are to us, then, you know, the more you might detect some of these things. So things like emotions, behaviour, but more about the sort of conscious behaviour, if you like. Memory, and then curiously, yeah, can't remember what other things I usually put in there. If I think of something else, learning generally, you know, the ability to sort of take in factual information. You know, other animals can't do that, and it tends to be located there. But I will probably have other things that will pop into my mind from what I should put on that list. It'll be something. All of a sudden, I'll remember what needs to go in there. Right, if we go down to the cerebellum, this bit here. This effectively stores, it's almost like a computer program, that it stores sequences of muscle contraction. This is a bit that controls voluntary movement. And there are reasons that we can't just do every motor activity perfectly immediately. You know, when we're born, we can't walk or do anything else much either. And we learn to walk. Now, part of that learning to walk is about strength, isn't it? Developing the strength in the legs. And it's partly about posture, because it's a need to get the spine upright, to push the bottom part of the spine forward, the top part of the spine backwards, and to actually get that sort of strength. Other than that, you know, you might have that. What else do you need to actually be able to walk? It's things like balance, isn't it? You know, and actually transferring the weight from one foot to the other and maintaining your balance while you transfer it onto the other. Our youngest grandson is, he was 1 in the middle of November, and he's not quite walking, which is very unusual for our family. We're, you know, usually walking around 10 or 11 months. But he was such an efficient crawler, he couldn't really see the point. I don't think he could get from A to B so fast on his hands and knees. But he is now just teetering on the brink. I got him to do 4 steps at the weekend, and he will walk if you hold his hands. He's off like anything, but it's about the cerebellum, and that's what you have to do sometimes, is to walk them, and so that the cerebellum is then actually recording the muscle contraction sequences. So I would expect in the next week or so, you know, if we just keep walking him round and round and round, the muscle, because he's walking, he's holding his own weight, and as he's doing that, that information is going to the cerebellum, it's being stored, and so before too long, he won't need that. It's probably just psychological security of hanging on to someone. He'll let go and he'll be off, and we'll wish we hadn't. But it has to be done, doesn't it? You know. But yes, it opens up a whole new range of naughty. But yeah, so what the cerebellum is doing, it's getting inputs from, say, the ears to do with balance, and from stretch receptors in tendons and things like that. And it's putting together this sequence of muscle contractions. So it controls just what we'd call voluntary movement, really. And, you know, if you're learning to do something physical, something like, you know, if you take on a sport, or, you know, something elaborate like gymnastics or Strictly Come Dancing, if any of you watch that, we do watch it, and I think it's absolutely phenomenal what they can learn to do in the time that they have. But what they're having to do is to get the cerebellum to store sequences of movements and muscle contractions until they're automatic and they don't have to think about them. Because we don't have to think about walking, do we? You know, so that's what's going on in there. Or, you know, even things like playing the piano and that sort of thing, you know, learning these automatic sort of sequences. So the cerebellum is doing that for us. Right, what else have we got? Well, there's a bit there that I've not really done very well here, called the brain stem, and I should have joined this bit up here. I have made a serious mess of that. I'll come back to it in a minute. However, up here we've got several areas I just want to identify. This sort of curvy thing here, it's called the corpus callosum. And these are fibres that connect the right and left hemispheres, nerve fibres that link the two sides, so that the left and right know what the other side is doing. Now, this is my sidetrack onto the difference between male and female brains. And whether this is true or not, I can't be 100% sure. But, you know, when you're reading things, you come across stuff that sticks in your mind. And I remember reading some years ago that it's been generally found that the connections between the two sides in women are denser than the connections in men. Whether that is true, I don't know. But it would account for that difference that a lot of us do identify. Perhaps I would say ability, but let's say the tendency of women to multitask, as opposed to the ability or tendency of men to focus and not want to be distracted by other things. And you can see how those actually, in a male-female relationship, those are actually really complementary skills, if you like, or abilities. Now, that's not to say that that's the only way things work, obviously, but if you take us back into our sort of early history, then male-female roles were fairly clearly defined, and men would go out hunting animals and would need to focus. All right, you can't be seeing a wild boar in the distance, go for it with your spear and suddenly be distracted by some berries or something like that, can you? It just, you know, you're not going to eat more than a few berries. On the other hand, the women would tend not to do that. They would stay back with the children, perhaps, and they'd have to watch the children and look for danger and get the berries and one or two other, you know, the attention would need to be in more places. So you can see how they would complement each other. Now we just tend to throw it at each other as insults, don't we? I multitask to the point of ridiculousness sometimes, and it's what my husband defines as the ability to do loads of things at the same time and none of them properly. You know, and that there is a tendency for that. You know, he'll say, you know, what's happened to such and such, or, you know, what about, why haven't you done the washing up? Or I see the washing up still in the sink. He wouldn't be as, yeah, he wouldn't take the risk of saying, why haven't you done it? But he said, oh, the washing up's still in the sink. And I would have run the water, left it in the sink, and then put the kettle on, then sat down at the computer to do something else while thinking about, you know, and there will be several things. And yes, the washing up got left until the water goes cold and he gets cross. But, you know, it is that, yeah, just not focusing. Whereas if he's focusing, let's say, watching something on the television and I speak to him, yeah, I'm in trouble. Yes. No, I'm watching this and I can't listen and answer at the same time. Whereas I will watch the television, play on the tablet, talk to somebody at the same time. You know, not, like I say, not do any of it properly, maybe, but then I won't remember what's been on the television, perhaps. And you know, but you know, between the two abilities, there's a huge scale. But the corpus callosum could well be responsible for at least part of that, couldn't it? You know, in terms of the two sides, either, you know, sending a lot of information both ways and trying to do everything at once, or so keeping most of it to one place, organise it, get on with it, focus on it, and that's, you know, the other option, isn't it? So that's my view on the corpus callosum and the the difference in brains. But it's, like I say, you know, it is a whole spectrum. It's not just one or the other. Underneath that, we've got an area called the thalamus and then the hypothalamus. I'm not going to label the I suppose I could. Thalamus is about, it's not a very good place for the thalamus, but it'll do. The thalamus is sort of like a switchboard, but it's sending nerve impulses to the right bits of the brain. And then you have a bit underneath it, hence it's called the hypothalamus. Right, hypo means under, doesn't it? And we've met the hypothalamus when we were doing hormones. We had that connection between the hypothalamus and the pituitary gland. So that little dangly do there, that's the pituitary. So that's where the nervous system and the endocrine system interact. We then have, my worst bit on here is what happened around here. And this is an area called the brain stem. Okay, now I'm going to end up putting these bits in the wrong order. It's just one of those things that I never get right, I don't think. There's two bits identified on your diagrams. We have the lowest part, well, there's three sections to it. It doesn't matter which is which on here. The bit that's nearest the brain I think they've not labelled the third part, but it's called the midbrain. And it's a fairly primitive bit, and it's like that area where spinal cord becomes brain. And there's some fairly primitive sort of responses there as well. You then get an area called the pons. Now, the pons, how good's your French? What's the French for bridge? It's pont, P-O-N-T, as opposed to P-O-N-S, but, you know, the basis is the same. And the pons is a bridge. It is a neuron bridge between the brain and the spinal cord. So I will just stick with the pons for now. So I'm not going to put the midbrain on there. So you've got the pons, which is a, let's say, bridge. So again, that's helping to direct impulses in the right direction. And then the bit that is most critical to our entire survival is this bit. And I have problems getting people to pronounce this properly. Right, those four letters there, D-U-L-L, how do you pronounce those? D-U-L-L is pronounced dull. It's a dull day today. Medulla. People do, medulla, yeah. Yeah, but if you think about it, that word, that bit in the middle is dull. It's medulla. It's, yeah, but a lot of people say medulla. And I've heard it on television as well. But yeah, the double L shortens the U. So the English lesson comes in as well. But medulla, yeah, like middle. You know, it's not that that's got the vowel in there, is it? Middle, right? Dulla. So medulla. Medulla just means middle. So actually, yes, possibly the midbrain is actually a bit below that, because medulla would sort of sound as though it's in the middle of those three. I'm not going to put the third bit in, partly because I can't remember where it is. But the medulla oblongata is the most important bit of the entire brain, because that's the bit that keeps us alive. And that's the bit that controls things like your heart and breathing rate. And in the unfortunate circumstance where somebody's suffered a severe brain injury, and they're looking at turning off a life support machine or something like that, and they look for brain death, that's the bit that they look for a response from. And families can find it quite disturbing, because you may still be able to get responses from the cerebrum. You know, if you might see flickering of the eyelids and things like that. But if the medulla isn't functioning, you can't maintain your own breathing and heart rate. So, you know, that really is when the control of the body has stopped, isn't it? So the medulla oblongata, the particular bits there, so controls heart and breathing. So that is a really critical bit. So I think that's not all it does particularly, but it has within it a respiratory centre for controlling the muscles that move the lung. They don't move the lungs, move the ribs and the diaphragm to enable us to breathe. And it has a cardiovascular centre, right? You'll need that if you're doing the blood pressure assignment, okay? Yet to be issued, a long way off yet. But the cardiovascular centre, the cardio bit, it controls the heart rate, the force, and the vascular bit, it controls the blood vessels and how narrow or wide they are. So, you know, without that, we've got no hope of controlling heart and breathing. So, yeah. That's the most critical bit of the brain. Right, let's have a, yeah, we'll have a look at the model here. I think I'll pause the recording while I do this. Okay, looking at your diagram in your handouts, then you've got most of those bits plus some others. If we start on the, where should we start? Let's start on the top right and work our way around clockwise. See if you can work those out. Top right would be, yes, it is the parietal lobe. So that one up there is like part of the roof, if you like. So parietal lobe is up there. Coming down the next one under that, it is the, corpus callosum. The labels sort of jump about a bit, so it's not too obvious. Next label down would be That's right, occipital lobe. That's the one at the back of the head, mostly to do with vision in there. Yes, cerebellum under that, the thing that looks like a peach. Followed by something I didn't put on mine. Yeah, spinal cord. Then if we come over the other side, we've got medulla, yeah. So medulla oblongata. Coming up that side, we have pons, followed by, it is the temporal lobe, yeah, it's pointing to that one that's sort of at the back of what you're looking at, so that's the 3D issue there. Followed by pituitary, yeah. And we haven't got the hypothalamus on this one, which is pituitary. And at the top, frontal lobe, yes. Right. Okay, I mean, the thing is, bear in mind, you know, you can make a whole degree out of this pretty much, can't you? And so we are touching it so superficially, you know, that we're not really covering very much that's of a lot of use. But, you know, apart from neurology, obviously, you've got psychology as the other side of how the brain works, haven't you? I mean, the neurology and neurobiology generally would look at the structure and function at the level of neurons and control of specific sensory and motor responses, perhaps. But, you know, you've got the psychology side that's all about things like memory and emotion and behaviour and addiction and behaviours generally. So, you know, in itself, brain science is huge, isn't it? So, yeah, this is the merest introduction to it. We did have a student a few years ago who went off to do a neuroscience degree. I can't remember where he went, Exeter, I think, which was really good. I don't know, no idea how he's getting on. Very often I don't hear from people. Curiously, yesterday when I was in Cambridge, I had just finished my meeting at Anglia Ruskin. I was going to cross the road to go back to the bus stop and somebody called my name. And it was two of last year's students. That was really quite strange to be in the middle of somewhere else and, you know, yeah, they're getting on fine. Right, moving on then. That was all we're going to do on the brain, really, is just, you know, where are different bits? What's their approximate function? The next bit is not in your handouts. The next thing you've got in your handouts is temperature control, and that's always a bit of a bonus item if I have time. And I think I'm probably not going to. What I do want to just do a little bit on is a little bit about reflexes and possibly a little bit on the autonomic nervous system. But reflexes, I think, are quite interesting, but I've given up asking about them in exam questions. The reason being, people find the diagrams really confusing with reflexes. So this is like a little sidetrack. But reflexes are almost invariably protective. And they protect us from falling over and damaging ourselves and things like that. And the thing that's common to reflexes is they don't involve any processing in the central nervous system. And they are composed of only 2 or 3 neurons, and that is it. So no more, all right? Different reflexes have 2 and others have 3. So 2 or 3 neurons only. I'm going to draw one with 3. So the reflex itself, reflexes generally don't involve central nervous system processing, but you do get to find out what the response has been. I'll tell you about that in a minute. So, for example, when I tipped boiling water over my hand the other week, which is now, yeah, remarkably better. What was my first response? Well, I didn't actually. Strangely enough, I didn't drop everything, and that's because it was food. Uh, and this is often the way. If you're holding something that's got your dinner in it, doesn't matter how hot it is, you will put it down. But there was a scream, and that was my first response. And it was to get away from the heat. But if you touch something hot, generally you will pull your hand away before you even know it's hot. All right. Similarly, if you stand on something sharp, I mean, the classic would be a drawing pin, but who has drawing pins around these days? I would say there's a ongoing competition here for the most painful thing to stand on. Yeah, Lego is one option, or a 3-pin plug the wrong way up is the other. Glass is pretty bad, but yeah, that will tend to dig itself in your foot and you know where you are. But Lego, it's those corners on Lego, isn't it? So. Sorry? Plug is the other one, yeah, with a 3-pin plug. Yeah, leave that the wrong way up and stand on that in the middle of the night. Oh, like the grip strip thing. Oh, well, if they're exposed, yeah, they shouldn't be. But yeah, if you stand on a grip strip for a carpet, then yeah, from around the edges, the little hooks that come up, yeah. There are some pretty nasty things to stand on. But your first response is always to pull your foot away, isn't it? Before you even realise what you've stood on. Sorry? Well, you could, yes, but the reflex is to get away from it. But actually, sometimes going past the point of pain can actually reduce it, can't it? And this is where reflexes come in. And then we have others, like if you shine a light into somebody's eyes, apart from them closing, what happens to the pupil? It gets smaller in bright light to protect the inside of your eye, but then in dull, lower light, it'll open up, it'll dilate. But yeah, you can't do anything about controlling that. And things like as we walk, we stretch, and this is one of the things about learning to walk. We stretch tendons, and that fires off impulses that give us a reflex that brings our lower leg forward. So when your leg is at the back of a step and your leg bends, that stretches the tendon just under your kneecap, and so your leg swings through so you can take the next step, doesn't it? Yeah. So think about walking. I'm not going to get on the desk to demonstrate, but you step, all right, we step forward, and then the back leg bends. As it bends at the back, the tendon under your kneecap is stretching, so your thigh muscle contracts and brings that leg forward. Because if it didn't, you would fall flat on your face, wouldn't you? So you need your leg to be there in front of you. That's the knee-jerk reflex that they test with a little hammer if they're checking out your nervous system. You cross your legs over, tap just below the kneecap, and your leg's supposed to kick forward. The thing is, if you're concentrating on it and you're looking at it, you probably won't because you can override that. But actually, you get those reflexes in all your joints. So if a doctor is concerned about your nervous system, they'll actually sort of tap other joints, and they can actually detect very, very small reflexes if they tap on the tendons. But the way this is usually drawn really confuses the heck out of people. So I've stopped putting this in the exam because so many people get it wrong. And it's because of the scale of things. If we were to start with, let's say we've got a flame, a Bunsen burner flame or something like that. And your finger, you think, oh, that's pretty, I'll poke my finger in it. Why would you? And now this is where things start to go out of scale. Here's a finger, right? Huge finger. Whoops. All right, fingernail. I don't know what happened there. Don't ask. I was concerned about that bit. You start off with sensory nerve endings in here. They transmit along a sensory neuron up your arm, and then you've got to assume, this is all the way up the arm and towards your spine, And as it gets to your spinal cord, it goes in a nerve at the side. I'm going to draw the outline of this in a minute, but I've got to carry on with this while I've got it. This is a sensory neuron, so it's got a cell body part way along the way. All right, and that's going into your spine. So, anybody would think I've drawn these before. This is your spine over here. So now you're thinking finger, spine. Yeah, and that's the problem that we have with these diagrams, that people just don't get it. All right, you've got to assume arm comes in here. Right, and so that's our sensory neuron. Now in the spinal cord, you do actually have a little connector neuron for this particular response. So it's just a quick one. And then you have a motor neuron that starts in there, comes out along a motor nerve from your spine and attaches itself to a muscle in your arm. All right, here's some muscle that produces a response. Okay? And that's all there is in that complete pathway. Now, you know, if you've touched something hot, your initial response then is to pull your hand away, because it reduces the damage, if nothing else. But you do get to know about it, so you can do other things. So luckily, you know, because I was pouring these, you know, the pan of carrots into the sink, luckily I could just put the pan down, turn the cold water on, and I think that's what saved an awful lot of damage. You know, and it was very lucky I was where I was. But clearly there are other responses. There's the shouting. There was, can't remember if there was swearing that day. No, there might have been, because I don't think we had any grandchildren around. So grandchildren filter was on. But yeah, and then there was the sort of, you know, waving it about, blowing on it, putting it under the tap, all the more elaborate sort of responses. So somewhere in there we've got to have a connection to the brain, but it's separate. It's not part of the reflex. So what you have here is your sensory neuron. You've then got your connector or relay neuron in the spine. So all we've got here, oops, I was going to write relay twice. All you've got is these three neurons and your motor neuron here, and that is it. And that's all that's required for you to do that. However, in order to let your brain know, to be a little bit, you know, aware, then there is a connection that goes to the brain. Whoops. So, you know, that'll take a little bit longer to get there, but it enables you to put together a more elaborate response, something that's a little bit more useful in terms of dealing with potential problems. But reflexes like pulling your hand away from something hot, or your foot away from something sharp, or you know, generally three neurons is not unusual. There are ones like I think the pupil reflex, I think, is only two, essentially into the brain, out of the brain. It doesn't even bother, you know, talking to anything in between. You know, it is so. The thing is, it needs to be a very rapid response. They all do. You know, the longer you leave it, you know, it could just be a few fractions of a second, and you know, you could have caused yourself serious damage. But there are other reflexes we learn. You know, you're probably aware. You know, we learn things like we learn to control certain reflexes as well. For example, at the base of the bladder, there are two sphincter muscles. Sphincter muscles are wonderful. We have got I don't know how many. We've got thousands probably. They open and control. They open and close tubes. Right, we know where one particular one is. The mouth. But it's better than that one. But that's a sphincter muscle. Your mouth. You go like that. You go like that. Circular muscle. Eyes as well. Actually, you know, screwing up your eyes. But yeah, the other one as well. But there are sphincters all over the place. There's one at the top and bottom end of the stomach, for example. And if the top one opens and your stomach contracts, then everything comes out. But if the bottom one opens, then that allows little bits of food to go through. Blood vessels have a lot of sphincter muscles. Where was I going with sphincters? Oh, bladder, sorry. Got sidetracked. all over the body, but where I was meant to be. The bladder's got an internal sphincter and external sphincter around its neck, and one of those is voluntary, the other one is not. The voluntary one is the one that we learn to control in our first couple of years of life. You know, we learn to keep it closed. Otherwise, when it's full, when the bladder stretches, it will just empty. But the other one, we don't learn control over. So there does come a point where if you try to keep control over your bladder, it will reach a point where you lose that control and it will just empty for safety, because a split bladder is a dangerous thing. And what stops the bladder being able to just keep filling, you have stretch receptors in the walls. So those baroreceptors we talked about, the pressure receptors in the walls of the bladder, will enable us to be aware of it filling up and then taking control of the emptying process. But on another sidetrack, a few years ago they were talking about a serious increase in the number of people ending up in accident and emergency due to ruptured bladders, and it's associated with binge drinking, because what tends to happen is that the nerve endings in the bladder wall become numbed by alcohol, because the alcohol has a sort of anaesthetic effect on the nerve endings, so the body is not detecting the stretch. And so even the reflex isn't working, and the bladder fills to the point where it actually ruptures. So it's, a really dangerous thing, that. Anyway, from ruptured bladders, this was only just this is a small sidetrack, because this involves the central nervous system, and it just adds a little bit more to, the brain we think of as being a very, very sort of controlled sort of, processing centre where information comes in. It's perhaps compared with things we already know, and it's, reasoned out perhaps, and then we formulate a response. Whereas these reflexes, they're the other side of the central nervous system, where it's just a very rapid sort of passing through. And lower animals rely an awful lot more on reflexes, don't they, than we do. Our learning ability has enabled us to develop much more elaborate behaviour. Okay, let's just do one final other bit that's, again, not going to get very much attention, and that is just to mention the autonomic nervous system, because that was central nervous system. We're going to do the eye next Monday, it'll be, all right, so starting here on Monday. And on Monday, we'll look at the eye sort of in total. I'll try and get the whole lot to fit into one session, I hope. That shouldn't be a problem. So we need to look at something to do with nerves, if we think back to our original structure of the nervous system. And so rather than choose body nerves, the somatic nerves, the autonomic nervous system is a bit more interesting in the sense that it works below the level of consciousness. Autonomic, that says, N-O-M. Autonomic nervous system. This is very much about homeostatic control. So what we've got going on here is two branches to it, or divisions they refer to. You have another reason for me doing this is that it will get a mention if you choose to do the blood pressure option in the assignment, because the sympathetic and parasympathetic control of heart rate are significant. So we have the sympathetic division, and these are all nerves that run alongside our somatic nerves. And this one functions like adrenaline. So it's sympathetic to adrenaline. So this one is very much about flight or fight. And so what it does is things like it raises the heart rate, it raises breathing rate, and it tends to divert blood, because we've only got enough blood to do so many things at a time. We can't run everything we want to at once. And it tends to divert blood away from the skin and the digestive system towards, what do you think for flight or fight? Muscles, yeah. Yeah. So diverts blood. So your skin goes pale, your heart rate goes up, your breathing rate goes up, yeah? So diverts blood from skin and digestion. Oh, and reproductive systems. So skin, digestive, repro systems to muscles. So, you know, if you think, you know, what you like when you're stressed, your heart rate will go up, your breathing rate will go up, your skin will go pale. Your digestive system feels a bit odd, doesn't it? You get the butterflies in your stomach sort of feeling, and you're really not up for that, right? But on the other hand, you know, you are actually ready to run away if you want to. The other, let's stick in the effect on the eyes. It actually dilates the pupils so that more light can get in. That could be a problem, you know. It dilates pupils. There are other things as well that. And it will connect with the adrenal glands to cause a production of adrenaline. But this is your initial response to shock, fear, surprise. It's the nervous system. And then if you need to maintain that, then the adrenaline kicks in. So, you know, you're lying in bed on your own in the house at night and you hear a noise downstairs, a crash. And it wakes you up and your heart rate's going like anything. So what happens next? Well, it sort of depends what you hear next, really, doesn't it? If you then hear, it's only me, or meow, or something, you're fine. You know what's caused it. You can go straight back to sleep again. It's not a problem because it was only your nervous system kicked off. If the next thing you hear is footsteps or cupboards being opened or something like that, you've then got to decide what you're going to do. You know, if you think you've got an intruder, what do you do? A flight or fight thing. I keep a big stick by my bedroom door. I have one of those hiking sticks. I got one of those big. I have a wooden hiking stick by the bedroom door because we've had several burglaries at night, and so I'm up for that. It's it sounds odd, but another sidetrack. I do a Tai Chi class once a week. I don't know if you all know what Tai Chi is. The Chinese exercises, they're really slow, what is actually a form of Kung Fu. But we do several other variations on it, which are actually all essentially martial arts. We do Tai Chi fan. So you do things with a fan and you think, fans? What are they? Yeah, well, these fans used to have metal spikes on the end and they'd be hidden up the Chinese big sleeve. You could actually kill someone with a fan. And we also do swords and we do Tai Chi stick. They're supposed to be that big, but that is why I keep my stick by the door, because I could use it, and I would. I discovered I would once. I didn't. It wasn't actually a person in the house, but I'd come back from somewhere on my own, and my son was in the house at the time. But as I came back, somebody came out of the side gate, and we both confronted this guy, and he was clearly looking to break in. Looking for a party or looking for so-and-so. We knew he wasn't, so we did get the police. But then my son went out and I was on my own as I went to bed, and I went round the entire outside of the house with my stick, and I was ready. So I knew I would as well. But yeah, and that, you know, why could I do that? Well, it wasn't just my sympathetic nervous system. That's where the adrenaline had kicked in. And needless to say, I didn't sleep particularly well that night, because once the adrenaline's kicked in, of course, adrenaline's a hormone. So it's circulating for ages, isn't it? And you then got to let it go down. You know, your body's got to metabolize it and take it out of your system before you can go back to sleep. So, this is the starter, the sympathetic nervous system, and if you need to maintain a stress level, then adrenaline and possibly other hormones kick off as well. But the other side of it is the parasympathetic. So if this one's flight or fight, the parasympathetic, they say, rest and digest. So the parasympathetic is our relaxation one. And these are a separate set of nerves, but you can tell what's happening here is that you've got a balance constantly, and this is homeostasis, isn't it? You know, up a bit, down a bit, one of the, you know, bit of this one, bit of that one, and so they're both kicking in at different times to give you the most appropriate sort of response. So the obvious thing here, it lowers your heart rate. Actually, I'll lump these together. Lowers heart and breathing rate, so you know you can relax and just get on with digesting. So lower heart and breathing rate. Let's do these in the same order. And then, of course, your blood can now go to your skin, digestive and repro systems. So blood, so you're relaxed, you know. So blood goes to skin, digestion, reproduction. So, you know, it's not just atmosphere, you know, your parasympathetic nervous system's got to be in the right frame of mind for that. And then it will tend to constrict the pupils, which is curious, actually, thinking about it. Actually, just thinking about the reproductive side of things. Because don't they say, you know, if you look at two identical faces, but with a difference in the dilation of the pupils, isn't the one with the dilated pupils supposed to look more attractive? I believe, which contradicts this, doesn't it, really, if you think about it, because somebody with constricted pupils is under the influence of their parasympathetic nervous system, so is more likely to be up for reproduction. So I don't know, but you know, there's a lot of psychology in there as well, isn't there? So yeah, I'm not sure you know what that's rooted in. But you know, we've got two systems working together below our level of consciousness, controlling all this homeostatic stuff. Okay, that's a good place to stop then. So I'll turn this off.